Hello guys, welcome to IB360. This is the second class on cells. The first class, Introduction to Cells, has already been uploaded. Click on the I button above and watch the full video first and then come and understand today's topic. Today's topic is prokaryotic cell. In the first class, we had talked about two superkingdoms. Cells are broadly classified into two superkingdoms, prokaryota and eukaryota, on the basis of their organization of DNA. Prokaryotes, they have incipient nucleus, underdeveloped. Their nuclear material can be described using words such as naked, circular, and double stranded. So we say dsDNA actually. Now this kind of nuclear material, since it is this nucleoid is one single chromosome, can also be called prochromosome. We have talked about all this in the previous class. It's just a quick recapitulation. Or genophore. Now per prokaryotic cell, there need not be only one nucleoid. There can be several copies of the authentic original nucleoid. Usually for schematic representation of bacterial cells, we show only one single nucleoid, but practically there are usually more than one copies of the genetic material. So it is obviously a duplex. It is not single stranded. In several questions we make this common mistake, but it is not single stranded genetic material. It is definitely a duplex, but it is circular. That means it is closed. It is complete and closed. And why naked? Because it lacks proteins like histones, and it lacks a nuclear covering. There is no nuclear covering. It is in direct contact with the cytosol, as you can see in this diagram. It is in direct contact with the cytosol. There is no nucleoplasm to protect it or to envelop it. So all these adjectives can very aptly describe a bacterial nuclear material. Then comes the cell wall. In the previous class, we pointed that roughly there are four types of organisms which can be categorized under prokaryota. One, bacteria; second, cyanobacteria or blue-green algae; third, mycoplasma; and fourth, PPLO or pleuropneumonia-like organisms. But a very important aspect of this information is that cell wall is not found in all of them. Cell wall is found only in bacteria and in cyanobacteria. Mycoplasma and PPLO, they lack cell wall. It is a very basic but very important information. Then you see flagella and femorrhea. Both are appendages protruding out of the bacterial cell. Both are appendages protruding out, but there their similarity ends. Flagella is strictly a locomotory organ, meant mainly for locomotion. But femorrhea, or sometimes we also involve another structure called pili. Obviously, they are again not exactly same, but their function is almost same. That is anchorage. In this diagram, these structures. They are femorrhea. They are not pili. Femorrhea are much more in number. Femorrhea, they are almost 200 to 400 in number, and they are bristle shaped structures. As you can point out in the schematic diagram, they are mainly bristle shaped structures, and their main purpose is to anchor the bacterial cell to solid surfaces. Pili, on the other hand, they are much more solid, thicker, and longer structures. They would be supposedly like this, and they would be 1 to 4 per cell, and that too not in they're not present in every type of bacterial cell, only in the F plus ones which have a fertility factor, and they are mainly meant for conjugation. That means they are main job is to transfer the genetic material from the F plus bacteria to the F minus ones. That is the main job, that is conjugation. But also pili act in anchoring one bacterial cell to another, so that is also a type of anchorage. Both of them, they do anchoring, but they are not at all involved in locomotion. So that is the basic difference between flagella and femora or pili. One more important information under this context is flagella of prokaryotes. They are very significantly different than those of eukaryotes. Eukaryotes, they have a 9 plus 2 organization, 9 plus 2 microtubular organization of flagella. So we often call them 11 stranded. We often call the eukaryotic flagella 11 stranded. But prokaryotic flagella is unistranded. And another significant information is they show rotatory movement. They have rotatory motors which are embedded inside the cell wall, mainly in this periplasmic space between cell membrane and the glycocalyx, and there these rotatory motors are found which enable rotation. This is the structure I'm talking about. Later on we will study these things, basal, body, hook and the flagellin proteins alone in much more details, but for now you should remember that they show rotatory motion, unlike eukaryotic flagella which show a whiplash sort of motion to and fro, and they're unistranded. So these are the first three important characters of a typical prokaryotic cell. And now let's see what comes next. So this is another important aspect, that is the prokaryotic DNA packing. We know that prokaryotic genetic material, they lack histone proteins, and so they are called naked. But packing is still happening. So what are the elements which are involved? The first thing, they are the nucleoid proteins. They are mainly polyamines. Second are the anchors, anchors, loop anchors, and third, RNA. These are the three elements involved in packing prokaryotic DNA. These are the loop anchors, and in a flower-like arrangement, they are packing the entire chromosome. This is one single chromosome, and in a zigzag fashion, the proteins, which are also called NAPs, non-associated proteins, they are in a way packing this duplex of DNA, prochromosome. This is the prochromosome. So this is how the packing is done. In fact, in this, or rather later on, we will see that mainly this is how the nucleoid is found, and with it the mesosome is associated like this. So this is the flower like arrangement, and at the bending region, at these regions only the anchor loops are mainly present, as you can see. So these are the three important structures important for DNA packaging. So these are the other characters of prokaryotic cell. Photosynthetic thylakoids, mainly found in autotrophs, autotrophic prokaryotes like cyanobacteria or purple or green bacteria. And since they lack membrane lined organelles, so obviously chloroplasts won't be found, but they have chromatophores which are involved with photosynthesis. These structures are found inside prokaryotic cell, chromatophores. And another important thing is thylakoids are not found in stacks. They are lying freely in the cytoplasm, freely, like free entities, individual entities. They are not found in sac, uh, like in plant cells. We know that these are like piles of coin, and this unit is the granum. But this is not the situation in prokaryotic cell. They are freely available in the cytoplasm. And next comes the vacuoles. Vacuoles mainly involve gas vacuoles. And gas vacuoles, they have two purpose. One is to protect the cells from harmful radiation, and secondly is regulate buoyancy. Now for aquatic photosynthetic bacteria, prokaryotes, the gas vacuoles, they help them in positioning their surface in such a fashion that they can avail the sunlight to the maximum capacity. So allowing them to float and align themselves a very in a very convenient position, the gas vacuoles, and also protection against harmful radiation. Ribosomes are present. They are 70S type. They can be also polyribosomes. They are the main site for translation, and they are freely available in the cytoplasm. As simple as that. And one more thing about vacuoles, these gas vacuoles are falling under a bigger category, that is, those are the inclusion bodies. Inclusion bodies are non-living structures present in the cytoplasm. And inclusion bodies mainly involve three structures. One are these gas vacuoles, which we have talked about already. Second are the inorganic substances. Now under this, a very interesting information is there. So usually we see that bacteria, now in the environment that they're growing, they take up the minerals or the inorganic substances which are present in their surrounding. One such bacteria is Aquaspirillum magnetotacticum. The name is quite bombastic, Aquaspirillum magnetotacticum. So this is a bacteria which contains magnetosomes. What are magnetosomes? Magnetosomes are nothing but vesicles which have magnetite in them. And it's a very brilliant feature in them. This enables the bacteria to orientate themselves along geometric line, geomagnetic line, more to be more precise. So they have magnetosomes, vesicles which are storing magnetite, and that is turning them magnetic, and they align in accordance to geomagnetic line with the help of these vesicles. The third inclusion body include food reserves. We have already talked about, or we have already learned we have already seen that in cyanobacteria there are alpha granules or cyanophysian starch, also lipid granules, or which are called beta granules. And in bacteria in general, starch is replaced by glycogen. Protein granules are also found. Carboxysomes are there which occur in photosynthetic forms. So these are the various inclusion bodies. Gas vacuoles are one of them. Inorganic substances, they can pick up magnetite granules, as in this example we have seen. They can pick sulfur granules. They can pick volatile granules. They can pick normal iron granules. So basically they will pick the things which are present in the surrounding, and this only enhance their ability to pick up different colors with basic dyes, and then they would be called metachromatic granules. So these are informations about inclusion bodies, quite important. Okay, one envelope system, because there are no membrane bound organelle. The one envelope which we know about bacteria is a cell membrane, actually not cell membrane, cell envelope, which involves glycocalyx, cell wall, and then cell membrane. So the one envelope which we mean, or which we know in prokaryote, is the cellular envelope, which is covering the entire cytosol. No other membrane. DNA content is pretty low compared to eukaryotes. Prokaryote DNA content is pretty low. Then transcription translation. This is another interesting aspect of the prokaryotic cell, that both of them mostly they occur simultaneously. Both of these phenomenon, they occur simultaneously. So this is a typical prokaryotic cell, and you see transcription is taking place and translation takes place simultaneously. But what is the norm as per eukaryotes? We know eukaryotes, they have a well defined nuclear membrane and they will have nuclear pores. inside the nucleus, from the DNA template, transcription will take place, and H and RNA will be formed, which is which will get processed to form the mature RNA. It will have a capping and a tailing, and it will form the mature RNA. Now this mature RNA will travel through these nuclear pores, travel to the cytosol. It will encounter some ribosomes, and there the translation will resume, or rather the translation will begin. So transcription has a different site, translation has a different site, but the situation is not similar or not same here. Both transcription and translation taking place in cytoplasm. No proper boundary system is present, so both are taking place simultaneously. Another important thing, because we know that they do not have introns. Introns are interrupting nucleotides, which are unnecessary, and during processing there is a process called splicing, where introns are eliminated, and that processed mRNA is a mature mRNA I was talking about. But bacterial cell or prokaryotic cell, they hardly have introns. They hardly have introns, so they do not have that requirement of splicing. They do not have that requirement for processing. So their mechanism, the central dogma of molecular theory, is much more oversimplified in them. Okay, now respiratory enzymes. Respiratory enzymes are found in mesosomes. We have to talk in a much more detailed fashion about mesosomes, and we will do that in the next class. So for today we have discussed these 11 basic characters of prokaryotic cell. We have talked about nuclear material, we have talked about cell wall, flagella fimbriae, the basic difference between flagella fimbriae and pili, DNA packing in prokaryotes, then the photosynthetic thylakoids where they are found, the vacuoles, the inclusion bodies, and lastly the transcription translocation thing and respiratory enzyme thing. So I hope you have enjoyed the video. If you like it, please hit the like button, share the video, and subscribe to our channel IB360. Till then, stay safe and stay tuned for my next video on cell. Thank you guys.